All right, Chapter 20, Genetics and Human Inheritance. In this chapter, we're going to cover the principles of inheritance, breaks in the chromosomes, and how to detect genetic disorders. We're going to start off by talking about how genetics is the study of both the stability of inheritance, the stability and the the variability of inheritance. So the stability of inheritance means that your parents were humans, you will be a human, and you are human, and your offspring will be human. But there's also a great deal of variation from one generation to the next. Prior to the work of the first true geneticist, Gregor Mendel, the concept of blended inheritance was already in place. This was the idea that parents of contrasting appearance would produce offspring of intermediate appearance, or the idea that if you bred a red flower with a white flower, you would always get a pink flower. However, this didn't always pan out when every few generations bright red or pure white flowers were being produced. This was just written off at the time as being a facet of the instability of the genetic material. This theory was so popular and widely spread until the work of Gregor Mendel. He is sometimes referred to as the father of modern genetics. His work did not agree with the concept of blended inheritance, so he proposed his own laws about genetic inheritance that we still use today. Gregor Mendel was a monk in the 19th century. He grew up in a region of what was then Austria, now part of the Czech Republic. He studied science and mathematics at the University of Vienna. He conducted breeding experiments with the garden pea Pisum sativum, and he ultimately studied how single genes are going to be inherited from parent to offspring. He used pea plants because pea plants were, one, super easy to grow, and two, had very discrete binary traits. And we're going to kind of talk about that. The plants were they were very objective to see. Plants were either going to be short or tall. They were either going to be green pea pods or yellow pea pods. They were very short, objective, and binary. Another great thing about the peas is that they are typically self-pollinating, so he could study purebred generations, but they are capable of crossbreeding, so he could intentionally and specifically crossbreed his plants. So he developed his laws from his crossbreeding experiments, meaning that he took plants that exhibited two different traits and he crossed them to see what the results would be. His crossbreeding in his experiments, he set up a few controls. Starting with the experimental control, he controlled the parents' original genetic information. He started off with parents that were initially purebred or true breeding, meaning that for many generations they had always only produced one specific genetic trait. He also chose varieties that only varied by one trait, meaning that they were identical in every way except for the one characteristic that he was studying. So they would all be the same height and size and color flower, but maybe one would produce green seeds and the other would produce yellow seeds. His experiments were also done using a genetic cross. He specifically looked at tall and short plants. So he would take the pollen from a tall pea plant and fertilize it with the pollen of a short pea plant. Then he took the pollen of a short pea plant and fertilized a tall pea plant. These first crosses, the tall crossed with the short, was known as the parental generation or the P generation. The resulting generation, which was called the first generation or the F1 generation. What he found is when he crossed a tall breeding true plant with a short breeding true plant, he got a tall plant generation. When he took that F1 generation, crossed it against itself, he got what he called the F2 generation, or the second offspring generation. This time he found that when he crossed an F1 generation plant against an F1 generation plant, he got a variety of tall and short plants. What we found is that when we cross a tall breeding true plant with a short breeding true plant, we end up with an entirely tall generation. But when you cross that generation against itself, you're going to find in a roughly 3-to-1 ratio, 3 tall plants for every one short plant. And he found that this was, interestingly enough, this was true. Whether you were looking at tall versus short, inflated versus constricted, round, wrinkled, yellow and green, axial or terminal flower positions, purple versus white flowers, or green versus yellow pea pods, he found that one trait always prevailed over the other in a roughly 3-to-1 ratio. This was very interesting. So from this work, at the time, Mendel called his factors hereditary units. We now know and recognize these factors to be genes today. Mendel came up with or discovered proposed his law of segregation, the idea that there are genetic traits that can be passed down in discrete units from one generation to the next. Just because you started off with a tall plant and crossed it with a short plant and received an entirely tall generation does not mean that that short generation is completely lost. We may see that short genetic trait reappear. So genes are going to be segments of DNA that code for a specific protein that will play a structural or a functional role in the cell. A trait is simply a characteristic that we can observe produced by the actions of one or more gene-directed proteins. Alleles are going to be different forms of a gene. They produce for different versions of the trait that they determine. So in humans, an example of this could be the gene for freckles. There's one gene, one allele will code for freckles, another allele will not. It'll code for no freckles. Just like in plants, one gene will code for a tall plant, one gene will code for a short plant. What we find is that these alleles can actually be found at the exact same location on different chromosomes. So here we have a homologous pair of chromosomes. Let's say that this is chromosome 1 from mom and this is chromosome 1 from dad. What we'll find is that in roughly the same location, maybe right here, we have the gene that codes for finger length. And so let's say that we get a gene for short fingers from mom and a gene for short fingers from dad. If these two genes are exactly the same, then we will say that the individual is homozygous. However, it could be that you get a trait for long fingers from mom and short fingers from dad. In this case, we'll say the individual is heterozygous. Homozygous individuals are individuals that have two copies of the same allele. Heterozygous individuals are individuals with different alleles of a given gene, meaning you have one tall and one short allele, or one green allele versus one yellow allele. The dominant allele is what we call the allele that is always expressed regardless of the alternate gene. What we find is that if you have an allele for a tall plant and an allele for short plant, you're going to see only a tall plant. If you have an allele for a short plant and another allele for a short plant, you will see the short plant. But in the event that we have a heterozygous individual, the dominant gene is always going to be expressed. When we are determining or when we are indicating dominant versus recessive genes, we do this by using an uppercase and a lowercase letter. So for example, if we want to talk about the genotype for height, we might say that tall is going to be capital T and short will be lowercase t. We are going to use big T, little t, and not big T, little s for tall versus short, to help indicate that we are talking about the same gene but a different allele. We're going to use the same letter, but either uppercase or lowercase. So a recessive gene is going to be when the effects of an allele are masked in the heterozygous condition. Genotype versus phenotype. Genotype is going to be the alleles that are present. This tells us what is the genetic composition of the individual. Phenotype, on the other hand, refers to the physical traits or the observable physical presence of the individual. So for example, an individual who has freckles might have two genotypes. They might either have homozygous dominant or homozygous or heterozygous. Their genotype will be homozygous dominant or heterozygous. Their phenotype will be freckled. In the case that we are looking at a widow's peak, if we're looking at an individual who has a straight hairline, their genotype would be homozygous recessive, or little w, that's the genotype. The phenotype is a straight hairline. So phenotype is what you observe. Genotype is the type of alleles that are going to be present. Spend some time, practice with the rest of these individuals. See if you can put under each picture, what is the genotype, what is the phenotype. Here, table 20.1 helps review some of those common terms in genetics that we have already mentioned. Gamete formation. Mendel performed a second series of crosses to help develop his law of independent assortment. Initially, he was looking at monohybrid crosses when he was looking at only one trait. So plants were absolutely identical in every single way, except for one was tall, one was short. When he was developing his law of segregation, he started to look at dihybrid crosses, meaning that plants differed in two traits. For example, he might have crossed a tall plant with a green pod against a short plant with a yellow pod. It's important to note that once again he used true breeding plants or purebred plants that only produce that particular phenotype. So for many, many, many generations going back, they always produced tall green pods or always produced short yellow pods. The law of segregation that he came up with was that during gamete formation, two alleles for each gene are going to separate as the homologous chromosomes move towards opposite ends of the cell during meiosis. Each chromosome is going to be inherited independent of the other chromosomes, following the law of independent assortment. The law of independent assortment says just because you are dominant for height does not necessarily mean you will be dominant for color. So just because you received your mom's hairline doesn't mean you're going to receive your mom's foot size. Maybe you got your mom's hairline, but you got your dad's big old feet. Maybe you've got your mom's fingers, but you got your dad's nose. So just because you're receiving the trait for one doesn't necessarily mean that you're going to always receive those same dominant traits. So we're going to take a look at that in meiosis. All right, so this diagram here, figure 20.6, is going to kind of flesh out or fill out that idea. And I'm actually going to let you take a look at this diagram on your own and come to me if you have any questions about independent assortment. We're going to move on to Punnett squares. Punnett squares are just going to be a matrix that's used to help predict the genetic makeup of offspring of individuals of particular genotypes. So the rows are going to represent the gametes of one possible parent, whereas the columns are going to represent the gametes of another parent. So in this case, we have a parent who is going to be homozygous recessive for the trait of no freckles, and we have a parent who is going to be homozygous dominant for the trait of freckles. And what we're going to find is when you cross these two parents, we are going to get, when you cross a big F with little f, you get big F, little f. You get, when you cross homozygous dominant with homozygous recessive, you're going to get a heterozygous individual. And we're going to see that for each one of these. So homozygous dominant, a homozygous recessive, we get a heterozygous individual. The boxes are going to ultimately represent possible combinations of gametes. We're going to spend a bit of time in our discussion groups going over Punnett squares. We have several labs that are going to be dedicated to lots of time on Punnett squares. So please spend some time looking at this in your textbook, and if you have any questions at all, come prepared to ask questions because we're going to be using a lot of Punnett squares in class. So we're going to look at, real quick, a monohybrid cross. A monohybrid cross is a cross in which both parents are heterozygous for one trait of interest. So in this case, we're going to be crossing big F little f against big F little f. This genetic ratio for the offspring is going to come out in a 1:2 to 1 ratio of homozygous dominant, heterozygous, and homozygous recessive. The phenotypic ratio for such offspring are going to be 3:1 freckles to without freckles. So that's going to be the end results of our monohybrid cross. But let's take a look at what that's going to actually look like as a diagram. So if we were going to cross, we've already looked at this particular cross for a homozygous dominant against a homozygous recessive. We're going to end up with an entirely heterozygous population. In the case that we're looking at a dihybrid cross where we're looking at two traits, so in this case we're looking at two traits of interest, we're looking at freckles and widow's peak. If we were to cross an individual who had freckles and a widow's peak and was heterozygous for those two traits against an individual who was heterozygous for freckles and a widow's peak, we are going to find some interesting ratios. We're going to find that all of these in blue, here, these are all going to be individuals who are freckled with a widow's peak. The individuals who are freckled with a straight hairline are going to be here in this sort of lighter peach color, and those with no freckles but a widow's peak are going to be over here in orange. And then finally, the individuals with no freckles and a straight hairline